Hello, students. So we're going to talk a little bit about partial pressures of gases. And then we're also going to talk about kind of where those partial pressures differ in the body. And that will get us to kind of the mechanism of oxygen loading and unloading. And then also kind of we'll get into how CO2 is handled in the body, which is very different. So again, thinking about pressure gradients, now we got to think about partial pressures of gases. So this is a bit of a different idea of the gases kind of moving through the cardiovascular system from lungs to tissue, from tissue to lungs, and around and around and around and again. And this will kind of take us through a loop, so we'll be able to figure out exactly why we need to pick up oxygen in the lungs and deliver it to the tissues, and vice versa with carbon dioxide. So let's go back and talk about air for just a second. And I really don't want to overcomplicate things and stress too much about numbers. Some people like numbers. So if it helps you, I'm going to show you where these numbers come from. If the numbers confuse you, again, the way that I want you to really think about this for an introductory anatomy and physiology course is just where is there higher concentrations of things and where are there lower concentration of things, and that's going to be able to predict how things move. So first, let's ask the question: what is air? Air is a mixture of multiple gases. And the sum of all of those partial pressures of gases is, going to give us a total pressure. So air in the atmosphere is not just oxygen and carbon dioxide. There's other things there. And in fact, the bulk of our air that we breathe actually is filled with nitrogen. And that's just, for us, that's a gas that it just takes up space. So it goes into our lungs, it comes out, we do nothing with it. Other organisms fix nitrogen and deal with that. That's more of a general biology topic. I'm not worried. We're focusing mostly on oxygen. So if you were curious what it makes up in the air that you breathe in the atmosphere is about 20.9% oxygen. And as the air comes into the lungs and mixes in the alveoli with all of our used up spent air, right, that percentage drops pretty quickly. Okay, and that is really less. That's something to kind of think about too with lung volumes. So you're breathing in your normal tidal volume, that's about 500 mils, that's what you kind of need. And then if you want to exercise and increase how much air that you're breathing, you can always bring more air in. That will help you get more oxygen. But it's important to note that there's a lot of dead space in the respiratory system. So we think about the lungs being stretched out and then coming back. But all of those little tubes that we're leading from our nasal cavity into our trachea, that's a really big tube. Right, we've got our right and left bronchi coming off, also pretty big tubes, and then smaller and smaller bronchi and bronchioles. All of those tubes actually stay open. They're very patent for airflow. And they make up what we call the dead space, because the stuff that's in the tubes on the way to the alveoli never really contributes to respiration. So you always have so much air in your respiratory system. Even if we did a forced exhalation and pushed it as much as we could out, there would still be air in our trachea, in our bronchioles, in all of those structures. So that's one of the reasons why as soon as you take a breath of nice, wonderful oxygenated atmospheric air, you're going to get a drop there right off of the bat, because you're mixing with all of the used up stuff that's in your body. Okay. There's a certain amount of water vapor in the air, and obviously in our body, we're made of a large percentage of water. We like to keep things nice and damp inside the lungs to keep all the tissues nice and hydrated and healthy and happy. So the water volume is going to increase, and that actually decreases the solubility of some of these gases and makes it challenging. And then CO2, the waste product, and as we'll find out, the CO2 is actually more important than the oxygen, right? It's the waste product that'll kill us and modulate our reflex to breathe. And you're going to build up CO2 much quicker than you're going to run out of O2. It is actually a very small percentage, but again, this is the thing, there's too much of it will definitely negatively affect your breathing mechanisms. And we can see inside the body, right, that percentage goes up. So these numbers that are in this column, you're going to see them on the next slide. Again, if you're a person that likes numbers, go with it. If you're a person that doesn't like these numbers, then just, where's the gradient? Big to small, right? High to low. Okay, so let's go ahead and start here. Let's go ahead and start with the atmospheric air, because that's what we were just doing on the last slide. Okay, so anytime you see a P in front of the symbol for oxygen or CO2, that's again giving you the partial pressure of that gas. Okay, and then the reason it's only a partial pressure, again, there's other things in there that we're not going to discuss, right? And it's going to change, the solubility of those gases are going to change as we move through the cardiovascular system, and it's also going to change based on the needs of the tissues and what's going on in the alveoli. So if we breathe in, right, air from the atmosphere has a relatively high concentration of O2 and a relatively low concentration of CO2. So let's just do the O2, and then we'll go around the second time for the CO2 so we don't get confused. So let's just follow the O2 with inspired air. Okay, so oxygen is nice and high. The second we go through all of our respiratory structures, so through the trachea, into the lungs, right, in there, we can see that the partial pressure of oxygen already drops because we're mixing with all of that dead space air. But we still have a pretty good concentration of oxygen here, okay, nice and high concentration. So there is going to be pressure for the oxygen to diffuse from the atmosphere into the alveoli. So they're going to cross that alveolar membrane and actually get into those alveolar capillaries. So now there's pressure to diffuse inside the capillaries. We have a very high concentration of O2. So here we go. We're going to follow these black arrows, and now we're in arterial blood. Obviously, we will go through the heart, right, through our pulmonary veins with a nice high oxygen concentration. And look, we've already dropped from 104 to 100. We're already losing some along the way. Okay, then we will go through our left side heart circulation. And we will head on down to our tissues that are in need. So let's go down to the tissues. Ooh, tissues aren't in a good place. Look, the PO2 in tissues is only 40 millimeters of mercury. The blood coming in, ooh, is 100 millimeters of mercury. So now we have a pressure gradient again that we have to worry about. Which way is oxygen going to diffuse from the tissue capillaries to the tissues? Well, it's going to diffuse out because the arterial blood coming in through the arterial side of the capillaries is 100, and the gradient is very steep. So we will unload some oxygen here from our blood, and then we'll be able to make our tissues happy, give them what they need. So then as we leave on the venous side, now we have a deficiency in oxygen, right? We know that we only have 40 millimeters of mercury here for a partial pressure of oxygen. And then we're gonna go through the right side of the heart, and then we'll go back to the lung to pick up more oxygen, right? And then as we're coming from an area where we only have 40 millimeters of mercury compared to 160 millimeters in the atmospheric air that we're breathing in. Okay, so now we have pressure here, again, to pick up more oxygen, for the oxygen to diffuse from the atmosphere across the alveolar membrane into the capillary, and then we can go around again. Okay, let's go ahead and start again. And this time we're going to focus on the CO2. So atmospheric CO2 is actually very low. It's 0.3% millimeters of mercury, so very, very low. As, again, we are inside the alveoli, right? We're mixing with all of that kind of used up air that's in the body. So the partial pressure of CO2 is actually 40 millimeters of mercury inside the alveoli. Okay, so now the gradient's kind of going the other way. Okay, so in this situation, we actually have more CO2 inside the alveoli than outside. So the pressure is for the CO2 to diffuse out of the capillary, across the alveolar membrane, and then out into the atmosphere. Let's keep moving through the cardiovascular system. Okay, so we have, right, our 40 millimeters of mercury here of CO2. And then we're going to move into the arterial blood. We'll go through the left side of the heart. Still 40 millimeters of mercury, no change there. And then we're going to head down to the tissues. The tissues have used their oxygen, they've metabolized, and they've actually increased their CO2 concentration. So it's going to be about 45 millimeters of mercury here. 45 is bigger than 40. So the pressure is for CO2 to diffuse out of the tissues and into the capillary bed on the venous side. So we're going to get rid of it. So we're going to go from having 40 millimeters of mercury in the blood. Now we're going to look more like the tissue. Now we're going to pick up that 45 millimeters of mercury here. So now we're in the venous blood. So now we're carrying away. We've gone up in our partial pressure here, all the way through the right side of the lung. I'm sorry, the right side of the heart. Okay, and then we're coming back into the alveoli. So again, partial pressure coming into the venous side of the pulmonary circulation here. I'm sorry, the arterial side of the pulmonary circulation through the pulmonary arteries. They are deoxygenated. What's going to happen here is, in fact, now the pressure is for CO2 to diffuse out of the arterial capillaries into the alveolar space, right, and then out into the atmosphere. So we'll go from 45, and then we're going to come back, right, to the number we had on the venous side, 40 millimeters of mercury. So we don't actually have big shifts. We're only going from like 40 to 45 with the CO2. You actually see a much bigger shift with the oxygen. But again, don't let that fool you. CO2 is actually really dangerous to build up in the system. So we don't want it to get really out of that 40-45 window. It's more important to get rid of the waste product, the CO2, than it is to modulate the oxygen quantity. All right, and then, there are really two things going on here. I kept talking about the capillary beds that were in and wrapping around each alveolus, and then you've got the alveolus itself. And we know that that alveolus is connected to a respiratory bronchiole, right? And then we've got all the tubes going on all the way up through the respiratory tree. So we're going to take a look at this thing here. Right, this is showing one single alveolus and the capillary bed there. Excuse me. So we've got one single alveolus and the capillary bed there. And, we can have kind of a mismatch. That would be a problem if we didn't have enough blood flow or enough of an airway open to properly do exchange. So there is a mechanism in our body making sure that ventilation and perfusion are appropriately coupled. So we want to make sure our airway is wide enough to let the right amount of air in and out so we can do exchange. And then we also have to make sure that our capillary bed has blood flowing through it quickly enough that it is going to be able to pick up and move the oxygen and carbon dioxide. So there's got to be a match. We can't have like a crummy little wilty capillary bed here and then a big puffed up alveolus, or you know, a kind of wilted alveolus and then like a very engorged capillary bed. We have to match ventilation and perfusion so that as the red blood cells are moving through the capillary bed, they have the proper amount of time for diffusion to happen. So this is why we have to couple ventilation and perfusion, and this is why we have to match them. If you need a refresher on the definition for perfusion, right, this is blood flow per like tissue weight, right? So certain tissues weigh more, certain tissues are more densely in need of blood flow. So we have to make sure that we're matching ventilation and perfusion here. Okay, so let's take a look. First at perfusion. So perfusion is primarily driven by oxygen concentration. So we need to make sure that blood is delivered, right, to areas of high O2 concentration in the lung. So we need to make sure that our arterioles are opened up big enough in the lung so that we can get as much oxygen to diffuse into those capillary beds as possible. So let's go ahead and look at some of these mismatches here. All right, let's say that we're perfusing too much. Let's say that we have too much blood flow and our capillary bed has blood just like whizzing through it. If the alveolus isn't opened up enough, as it doesn't appear to be in this particular image, right, that's going to create a situation where oxygen is going to drop later in the tissues because the blood was just speeding through the cardiovascular system and didn't slow down enough. It'd be like if the SEPTA train was coming and it didn't come to a complete stop and they're like, hurry up, jump, get on. Not everyone's going to make it and get on the train. So we need to make sure the train is running through the station at an appropriate speed to pick up the oxygen. Okay, so if the alveolar oxygen is high, those arterioles will dilate so that they can have a better chance of picking up more oxygen. If alveolar O2 is low, the arterioles will constrict. So let's say this is a situation here where we didn't have adequate airflow and we had this big old capillary bed that was very vasodilated. We can simply adjust and match here. What we'll do is kind of constrict part of that capillary bed with some precapillary sphincters. We'll shut off a little bit of the blood flow so that there's less blood moving through there or it's moving at a slower rate, and then we can match up our ventilation and our perfusion. Okay, the other side of this is we could get the alveolus to actually fill up with a little bit more air if we get our respiratory bronchioles to not vasodilate, but bronchodilate, right? So if we get those airways to open up a little bit more, we can get more, not so much more in because we're actually more worried about CO2 here. So we can get more CO2 out. And again, we need to match that. So if CO2 starts to build up, let's say we are breath holding or we are exercising, or maybe we're in a contest with one of our friends rebreathing air into a paper bag, whatever you want to do. CO2 will start to build up and we got to get it out of there. So that's going to help and stimulate the bronchioles to open up and help us get the CO2 rich air, which is problematic for us, out of the body. So we can look at a situation here, and perhaps, right, we've got a problem now. We've got a very dilated bronchiole, trying to get CO2 out, having trouble getting it out. So we are going to bronchodilate, and then here the little capillary bed was all wilted and shriveled up from the scenario before. So, right, we need to get that kind of matching and moving in the right direction so we can get the CO2 out of the body. So what's going on the whole capillary bed is pretty much O2 driven. What is going on at the level of the alveolus is more CO2 driven. And they'll kind of seesaw back and forth as need be so that ventilation and perfusion are appropriately matched, so that when, right, the train, which is the circulatory system, and the red blood cells, you know, really moving through it, when the red blood cells arrive in the station, right, there is enough time for the air, specifically oxygen and carbon dioxide, to load and unload. Okay, so let's talk a little bit more specifically about what's going on with the red blood cells. So we've got an idea of how blood is moving through the cardiovascular system and where oxygen and carbon dioxide are getting picked up and dropped off. Now specifically, like, how is that carried? What is the mechanism? So we need to go back to the blood lecture and think a little bit about what's going on here. So oxygen is really transported in two ways in the body. One, you can dissolve a gas into a liquid. You certainly know that every time you open up a carbonated beverage, you know, whenever you open up the top and the bubbles come up, you certainly can put some gas into a liquid. You can only put 1.5% of the oxygen in your body into the liquid that is blood. It's not very much. Most 98.5% of the oxygen that is carried in the blood from the alveoli to the tissues and then back from the tissues back up to the alveoli is bound to hemoglobin, which is our protein that we find that is just packed into red blood cells. So you remember this thing, right? Its quaternary structure has 4 chains. Each chain contains a heme molecule, this little pizza thing with a cherry on top. And in the dead center of that heme molecule, right, there's a central iron atom. And that central iron atom is responsible for binding a single O2 molecule. So each individual oxyhemoglobin molecule can carry 4 O2 molecules. And we say that it is completely saturated whenever it is carrying all 4 O2 molecules, and then it is free to drop those off of the tissues and then come back to the lung and pick up more. Deoxyhemoglobin actually experiences a color shift as you unload the oxygen. That causes a shape change in the heme molecule, and that will indeed lead to a color change, which is kind of cool. So that's why venous blood is actually a much darker red. If you look at your veins, it looks blue. That's kind of like a color trick with the way that the lights are refracting through the skin. But there is some differences there between oxyhemoglobin and deoxy. So if a fully saturated hemoglobin molecule that is carrying 4 molecules of oxygen, right, that's what we're going to start kind of thinking about here. Okay. So we would say an oxygen or a hemoglobin molecule is fully saturated with oxygen 100% if all 4 heme groups are carrying O2. That's it. It is considered partially saturated any time that you are carrying between 1 and 3 hemoglobin or O2 molecules. And we're going to look at the curve of how oxygen is loaded and unloaded, but know that there are other things here that can kind of change the shape of that curve and change the pressure for loading or unloading. what we actually find out is that we don't completely unload the hemoglobin molecule when we go to the tissues. Even though we're going from like a partial pressure of 100 millimeters of mercury of oxygen down to like 40 in the tissues, we still often only dump off like 1 oxygen molecule, and then we take the 3 back up to the lungs. Seems kind of silly. But if we get in a really stressful situation, let's say low O2, maybe we're climbing a mountain and we're exercising, our body temperature goes up when we're exercising, blood pH starts to change and become a little bit more acidic, or our CO2 concentration goes up, that can actually get us into a space where maybe we are dropping off not just 1 oxygen molecule in the tissues, but 2 or maybe 3. It's really weird to get all 4 of them off of there. Typically doesn't happen. Okay, so let's look at our oxyhemoglobin dissociation curve. So this monster right here. It is a sigmoid shaped curve, which it's got like this kind of little kinky S bend in it. So we're kind of focusing our attention on that. And then we're looking at the y-axis. So this is showing the percent O2 saturation. Again, there's 4 molecules, so it's like 0, 25, 50, 75, 100. That's, you know, really where this has to go. And then, right, we can see the partial pressure, right, of the oxygen here on the x-axis. So basically where you're living, if you're doing normal quiet breathing and you're loading and unloading, where you're living on this curve is up here at 100% hemoglobin saturation whenever you're in the lung. And then we're looking at, this figure is really great. You can see the 4 little O2 molecules down there. Where you're living on the curve is really from here to here. If you're doing normal quiet breathing, so 100% saturated, 4 hemoglobin molecules in the lung, take a trip round to the tissues, and then whenever we're in the tissues and O2 is low, only 40 millimeters of mercury, we still only slip down to about 75% saturation at the tissues. So again, that means we've only let 1 little oxygen molecule off at the tissue, and that has done enough to make the tissue happy at rest. So again, we think of arterial blood being around 95 to 98% saturated. Again, we're not perfect. We can't be at 100, but we're close. So we're approaching 100% saturation in the arterial blood. And then again, if we're only taking off 1 O2 molecule, that should get us to about 75. We're not perfect, so we'll say 70. And that's where we're living on the curve. So just loading up to 4 O2 molecules and then dumping 1 of those off at the tissue, and then picking 1 back up in the tissue and then dropping it off again. All right, that was just sitting around resting, having a nice day. If we start to exercise, body temperature is going to go up. Our pH is going to start to drop. We're going to become more acidic. And that's actually linked to CO2 as well. So CO2 is going to build up. And then there's another thing called bisphosphoglycerate, which I will get back to in a minute. Okay, and what's going to go on with that is this is going to help shift our curve. So let's just go ahead and focus on the temperature curve. So this is the oxygen unloading curve. Again, remember percent saturation hemoglobin. We just don't have the cute little figure with showing the molecules there and the PO2. So focus on the purple curve, this one first. So that one's going to be a normal body temperature. So if the body temperature goes up, this is going to shift the curve in this particular direction. Okay, we still have that sigmoid curve, so it's still pretty flat there. But again, if we're trying to think about percent saturated hemoglobin, where we want to keep like the PO2 in the tissues, right? Those sorts of things are really going to affect what's going on here. So let's say if we're in the tissues, let's go up from 40% saturation. right? Again, if we were at a normal body temperature, that's hitting us on the curve right where we thought we would be, right? Again, and about 70–75% saturation with hemoglobin. But now if we then focus our attention on the red curve, we see that, oh, we're kind of dipping down here. So we might have to unload, if our body temperature goes up, we might have to unload 2 hemoglobin molecules and let 2 oxygens come off of those and get us down to, right, maybe more of a 50%. It's not going to be even, like one will give up 1 molecule of oxygen, another one will give up 2, so it's not going to be quite even. So as we shift to a higher temperature, presumably during exercise, and these other pressures are happening, we will unload, right, a second and possibly even more hemoglobin molecules, or sorry, oxygen molecules from the hemoglobin. pH, same thing. We're going to find out that there's actually a pretty big link between blood pH and blood CO2 in the next mechanism. So again, if we focus on the purple curve, a normal blood pH is 7.4 in a healthy human. Once you start exercising, your blood actually becomes a little bit more acidic. So that will drop your pH, and we'll see, again, we're kind of moving with this shift in this direction on the curve. So that mechanism is really cool in that as you start to need more oxygen in your tissues, things like CO2, temperature, pH will help unload a little bit more oxygen off of your hemoglobin molecules. Bisphosphoglycerate, that is a compound that you will see and encounter if you're kind of challenging the system with altitude. So that will help unload oxygen in a very similar way if you travel from sea level to altitude. That compound will help enact that mechanism to ensure oxygen unloading, just because now the air is thinner. Okay, let's take a little break before we get into how CO2 is transported in the blood.